Hi, I'm Randy Nesse from the Arizona State University Center for Evolution Medicine, and my guest today is Lee Nelson from University of Washington. She has actually been a practicing physician, a professor of medicine, and she also has a laboratory at the Hutchinson Research Institute. Welcome. She's here today to give a talk about microchimerism, which turns out to have all kinds of interesting ramifications. What is it? What is microchimerism?
So microchimerism is, the word derives from the chimera, which was a mythological creature that was part lion and part goat and part serpent. Micro just means small, and so it's a small amount of cells or DNA that came from a genetically disparate individual. So that's what the term means in medicine.
So a baby's cells that continue to circulate in the mother after birth?
Yes, or cells of origin in the baby, because those cells probably age. So if it's 10 years later.
Wait, 10 years later? The cells can still be in the mother 10 years later?
They can, 10, 20, 30,
Really?
probably indefinitely, yes.
Do they just live, or do they divide in the mother, or what?
Well, people don't really know exactly what they do, but you have to kind of assume that stem cells pass over, because you can detect these cells in all kinds of different cell population decades later.
So how was this discovered?
Well, the first report, and the person that really put this in the presence of the scientific and public eye, was Diana Bianchi's in 1996, where she described long-term persistence of male cells that were progenitor cells in women that had given birth to sons.
So I'll bet she looked under the microscope and thought, those are male cells, it must be a mistake. And then?
We'd have to ask her that. I didn't ask her that specific question.
Yeah.
But I know she had a very hard time getting the paper published.
Really?
She had
Everybody
to
said it's impossible.
Yeah.
Typical for big discoveries,
Yeah.
right?
Yeah.
You've been working on this for a long time now.
Yes.
How did you get into it?
And well, we kind of described the other side of it. We asked the question whether mother cells persist in everyone else. Mm-hmm. And this had been studied, interestingly, in immune deficiency, but no one had asked the control question. You know, what about non-immune deficient people? Do mother cells persist?
So how many of us have our mother cells still circulating in us?
It's probably universal, but
Really?
we can't say that scientifically, because at the limit of our ability to detect in humans, we don't detect it in everybody.
So what kind of cells are these? All cells in the body, or just certain blood cells?
Well, that was the next part of the story that was really interesting. I think in the beginning people thought, well, these must be interloper cells, they must be circulating blood cells. But in fact, what Ann Stevens showed, she's a pediatric rheumatologist, mm-hmm. who looked in the hearts of some babies that had a syndrome called neonatal lupus. And she found that most of the mother cells that were in those babies' hearts were actually cardiac myocytes. They were muscle cells. Mm-hmm. So clearly these cells can adopt the phenotype of a lot of different tissues. They can be a liver cell, they can be a beating heart cell, they can be a renal tubular cell, they can be a variety of different epithelial cells.
So these cells from the mother go into the baby and turn into actual tissues?
It looks that way, yes.
Wow.
Yeah.
Fascinating.
Yeah.